Well, thank you very much, Marina. Thanks, Veronica, too. And thanks, Stefan, I believe. Sven. Stefan. Thank you too, Sven. I would like to talk a little about my scientific work, but I realize this is an audience with mixed backgrounds. So I'll tell you, in large part, how I came to be doing what I'm doing and how one thing led to another, what was the process. And I'll tell you in a little detail about what the discoveries were also. I can begin just with a bit of family background. I should tell you, Freiburg is the first European city I ever visited when I was 15 years old. And I've been back here every few years, I would say. It's really one of my favorite cities in Europe, and it's a pleasure to be here. I'll start with that. You might guess from my name that my family came from Germany. My father was born in Berlin, and these were my grandparents who also lived in Berlin. Both of them were physicians. My grandmother was a pediatrician. My grandfather, on my father's side, was an internist, had some specific interest in cardiology. Neither of them were really scientists, but they had a general interest in science. My father was a scientist, really a very redoubtable scientist, outstanding scientist, also a clinician. This is how he looked at the age of 2 before the family had left Germany. And at the age of 7 in 1935, they moved to the United States, and my father grew up in Milwaukee, Wisconsin. He was a very bright student, and he was sent at the age of 15 to the University of Chicago, where there was a special program in which you could enroll as an undergraduate, go through all of your undergraduate work, and then through medical school, which normally took 8 years, but it was a 6-year program. And that's what he did, and he graduated from the University of Chicago with a bachelor's degree and an MD degree at the age of 21. He was the youngest student in his class, and he was also the valedictorian of his class. In the upper panel, you see how he looked on his graduation day, and just a few days later he got married to my mother, Bonnie Beutler, then Bonnie Fleischer. That was June 15, 1950. My mother had been the child of immigrant parents also. Her parents both came from Kiev sometime around the turn of the 20th century, and I don't really know all of the circumstances of their voyage to the United States. I do know that the family settled first in Chicago, but then my mother's immediate family moved to Tulsa, Oklahoma, and this is the house where she grew up. She later went back to the University of Chicago, where she majored in mathematics. After a few years in Chicago, in the year 1959, when I was about 2 years old, my family moved to California. So I grew up in Southern California, and my earliest memories focused on nature in large part. I was really interested in nature as a child, and as a teenager I used to go hiking in the San Gabriel Mountains. I especially liked to go on the Mount Wilson Trail. My own hometown was Arcadia. It was next to Sierra Madre, where you had access to the trail, and if you walked about 8 miles, you would rise 1 mile in altitude, and that would be the top of Mount Wilson, where there was a famous observatory that was important in making the theory of the expanding universe, measurements of the red shift. That wasn't so interesting to me, though. I liked the world of animals and plants more, especially animals. I remember trips to Sequoia National Park to see the redwoods, to Yosemite, also to the Grand Canyon. These were formative, I think, in my development. I was especially interested in birds, and I had a friend named Louis Bevere who actually did go on to become a professional ornithologist. At the age of 11 or 12, we were going bird watching, bird banding with real professionals, and I began photographing birds. Here you see some later photographs when my equipment got a little bit more elaborate, but it's something that I still do as a hobby. I always been interested in evolution, and I like to look at birds just for aesthetic reasons, and also because I find it amazing they've become so diversified in how they look and how they behave. But I was curious about life from an early age. I thought to myself, why is it that molecules sometimes arrange themselves to be part of living organisms? How is it that they're captured in that state? And how is it that after they are so co-opted, there is replication and you have more non-living matter incorporated into living things? Extremely interesting. My brother, Steve, my oldest brother, 6 years older than I, he went to University of California at San Diego, where I later went. And when I was about 14, he came home with one of his textbooks that he had finished with, and I was discussing these things with him, and he gave it to me. It was a book by James Watson, the second edition of Molecular Biology of the Gene. And I thought, that's exactly what I'm interested in, from what little I knew. This was an engaging book, and I read it all really rather quickly, but in all detail, every word of it. It began with a consideration of hydrogen bonding, how it is that water molecules especially attract one another, and what an interesting substance water is. And then it talked about how hydrogen bonds also maintain the conformation of proteins, leading to their overall shape. Then, of course, came the question of how proteins are made. They're a string of amino acids, and that particular sequence of amino acids is coded for in the genome, and there is a genetic code, a triplet code, whereby amino acids are made. First, the genetic code is read by transcription to make mRNA, and the mRNA is translated into proteins on ribosomes. So all this made sense to me, and I thought it was very, very interesting. And nonetheless, I hadn't worked in a laboratory to that time. I'd only been going around photographing birds and banding birds and marveling at nature. But at some point, my father, who was a real scientist, said to me that merely observing nature as you're doing, Bruce, that's not really science, and wouldn't you be better off if you came and worked in my laboratory? And I will pay you $1 per day, he said. And eventually I decided I didn't really need that. But he said you have to ask questions about a biological system if you want to be a real scientist, and you have to, through experimentation, answer these questions. Now, my father at that time would have looked about like in the middle picture here. He worked at the City of Hope Medical Center at that time. His discoveries to that point were mostly in the area of red cell enzymology. He had found that people with hemolytic anemia who had their red cells break spontaneously often had defects of certain enzymes that maintained the oxidation state of the red cell. He had made a really exceptional discovery when he was very young, in his 20s and early 30s. He had discovered that the disease favism, as it's called, also primaquine sensitivity, a kind of hemolytic anemia that was induced by consuming foods that led to oxidative metabolites or drugs that did so. This was caused by glucose 6-phosphate dehydrogenase deficiency. And it happened that this was an X-linked enzyme, and so males have only one copy of the gene and females have 2 copies. And he wondered about why it is that females don't have higher levels of the enzyme than males. This was something called dosage compensation that applies to all X-linked genes, and here he had one of particular interest. And he showed that in women, either one X chromosome or the other one was active, never both. And he concluded that there must be a shutoff of one of the X chromosomes early in embryogenesis. And that's actually how things are. He determined this by looking at the stability of glutathione in red cells, and he found the curve was biphasic in women who were heterozygous for G6PD deficiency, had one good copy of the gene, one bad copy. This was really the best, I think, in the field of epigenetics to that time. He discovered that a whole chromosome gets epigenetically silenced, and it made him quite famous throughout the world. This was simultaneously discovered by a woman named Mary Lyon, who worked with coat color genes in mice. In any case, I began working in his lab in 1972, and there I learned how to prepare proteins, how to analyze them, how to isolate them, and this was something that was useful to me later on. Repeatedly in those early years, I worked in different laboratories, and learning is quite subliminal, but I've tried to identify the points at which I learned things that were really important for me to succeed in the future, and definitely this was one of them. I got quite comfortable at purifying proteins by gel filtration, by isoelectric focusing, by electrophoresis, all the methods that were current at that time. I began by assaying enzymes in the lab, and this was done by a kind of linked biochemistry where ultimately you'd measure a substance called NADH, or alternatively NADPH, by looking at absorption in the ultraviolet region of the spectrum. My father developed these assays, and we used this piece of instrumentation, which now I have to say looks like a piece of junk, but at the time it was a really sophisticated piece of equipment. I didn't only work in my dad's lab, but in other labs as well. With Dan Lindsley in college a year or two later, I learned how one maps genes to chromosomes between visible phenotypic markers. Wasn't so interesting to me at the time. I did it with a red cell enzyme, actually, that I was interested in. I thought we could look at that electrophoretically and map it to a chromosome in the fly, and I was able to do that. And I understood how you establish a critical region and say that the mutation must be downstream from this marker and upstream from this marker, and therefore I know it's at this point on the chromosome. But you couldn't go any further in those days, and so I didn't quite understand the power of genetics. I thought that's very nice and interesting and theoretical, but I didn't see how it could be put to use. This gentleman in the middle, Abe Braudy, was the person from whom I first heard the word endotoxin, which I learned was lipopolysaccharide, and it became such a big part of my life later on. But at the time, Abe Braudy was interested in LPS because he thought maybe by passive immunization, by giving people antibodies against LPS, he could block endotoxic shock. So quite precociously I learned about LPS. Susumu Ono was a mammalian geneticist who had established that one of the X chromosomes becomes condensed. And he was a close colleague and friend of my father until he died a few years before my father did. He taught me a lot about mouse genetics and also about immunology as it stood at that time. So you see, I was building up a kind of knowledge that would serve me later on. I graduated from college, and then the time came to decide whether to go to medical school or to graduate school. And my dad said, definitely go to medical school, I guess because he had done so. And I said, what about an MD and a PhD degree? And he said, no, don't bother with that. If you're not just Mr. Beutler, then you'll be able to compete for grants, and you won't have any trouble getting funding, and you'll be able to do whatever you like. He told me that if I understood pathology and physiology, and really the mechanics of how a single organism, that being Homo sapiens, works, then you'll really have a basis to do experiments in many different areas. And that had apparently worked for him, and it certainly wound up working for me. In the course of being at the University of Chicago, we had to take a class in statistics. And my teacher was none other than Paul Meyer, whom many of you have heard of because he developed the Kaplan-Meier plot, in which one puts time into an accounting of survival, for example. And I had never really thought about statistics before, and they too became very important to me. And I had some contact with Paul Meyer, a lot of one-on-one discussion with him, and I learned from him the importance of blinded studies, double-blinded studies in particular, and not to be casual about that. And I also learned something that really stuck with me. I had never heard of R.A. Fisher, the famous statistician, and he introduced me to the concept of the null hypothesis, not Fisher, but Paul Meyer. And Specifically, R.A. Fisher said of the null hypothesis that it's never proved or established, but can be disproved. And he wrote, every experiment may be said to exist only in order to give the facts a chance of disproving the null hypothesis. And that too, probably I didn't quite understand at the time, but later it made great sense to me. And you'll be impressed at the number of null hypotheses we've come to test and how it's central to everything we do these days. I finished medical school. I decided that I would do a really tough residency if I could, because I knew I didn't want to go on and be a clinician in the long run. I was already eager to go back to being in the laboratory. But I decided, since it would be only for a couple of years, I would go to UT Southwestern. And there I did an internship in internal medicine, which was really quite exhausting, kind of at the limit of what a human being can stand from my point of view. And another year of neurology. And those two were useful to me. I really saw what a whole organism can tolerate, how to fix things when they go wrong, and I came to understand how to deal with most kinds of emergencies that I ever would need to. But things seemed repetitive to me. I didn't like medicine from the standpoint that it always involved a diagnosis and then usually quite a rote series of things to do, and I wanted to be in science again. And that brought me to leave and go to New York City, and I worked in the Rockefeller University in the Cerami laboratory, where there was a question about whether the immune system might contribute to wasting in chronic disease. And the classical example was, you look at an animal with trypanosomiasis, that animal will undergo severe wasting, develops anorexia, won't eat, usually loses most of its body weight, and yet the mass of parasites that are present is quite small, maybe only a few grams in an animal the size of a cow. How is the signal amplified, and how does it communicate with the energy storage tissues of the host? The model that was developed in that laboratory was to take endotoxin, to treat macrophages with endotoxin, and then to measure an activity that we called cachectin. Cachectin was defined by its ability, when placed on fat cells, to shut off their expression of lipoprotein lipase, which is an enzyme needed for fat cells to take up fatty acid chains. I went back to my roots in my father's lab, mentally speaking, and I decided I could purify this activity, no problem, and I worked on it. And rather quickly I did purify cachectin to homogeneity. I did so using a macrophage line called RAW264.7. I would treat those cells with LPS, use this assay to measure cachectin on fat cells, and I went through a series of steps and enriched it more and more until I had a single protein. And that protein I sequenced, and not long afterward I found that the sequence was very similar to human tumor necrosis factor, which had been isolated contemporaneously by people at Genentech. Now the workers at Genentech did not know something that I knew. They knew that this was a protein that would induce lysis of cancer cells in vitro and could cure animals, as Marina mentioned, of tumors that were grown in an experimental situation. But they didn't realize how toxic it was. And I, for my part, had begun to think that this was a mediator of endotoxin effects, since it carried out two very disparate kinds of effects in animals. And I thought maybe it's responsible for endotoxin-induced shock. I gave purified TNF to mouse TNF to mice, and I found that they looked very sick. If I gave more than 20 microgram, they would usually die, and they looked very much like animals that had been treated with LPS. I raised an antibody against TNF and passively immunized mice against their own TNF, and then treated them with LPS. And I showed that their survival improved significantly compared to animals that had been given just a control antibody. And that really convinced me that it was a mediator, although not the only mediator of endotoxic shock. I went a bit further and I identified receptors and made a receptor competition assay involving these molecules. I didn't clone them, but I used them to measure the amount of TNF in different biological fluids. And later I decided that I would use the receptor to make a highly specific, non-antigenic and stable inhibitor of TNF for therapeutic use. This I achieved together with Karsten Peppel and David Crawford, who worked in my lab at that time. And we fused the receptor to an immunoglobulin heavy chain. It was a kind of an antibody chimera, but the binding domain was the receptor, so it would bind only to TNF trimers. It would neutralize them and prevent them from binding to cells. And still 30 years later, this molecule is used clinically to treat rheumatoid arthritis, ankylosing spondylitis, psoriasis, other diseases. It had a major medical impact. But during this same time window, I was thinking more and more about how TNF biosynthesis is controlled. And I identified a transcriptional level of control, a translational level of control, and I realized all along really what I'm looking at is endotoxin signaling. And I realized the receptor for endotoxin wasn't known. And I thought, this is very strange. It's been nearly 100 years now since endotoxin, or LPS, was named as a substance, and nobody knows its receptor, and it's so important. And might it tell us how we sense microbes in the very first minutes after we encounter them? And I became more and more obsessed by this and decided this was what I really wanted to work on. LPS, I should mention, not only causes this kind of severe systemic inflammatory response, but it acts as an adjuvant. It has a radioprotective effect. It gives non-specific resistance to all kinds of infections. And these all are aspects of the same thing. In other words, they're properties of the innate immune response. Insofar as an innate immune response really is what we call macroscopically an inflammatory response, I thought maybe all inflammation is initiated by this type of receptor, and that too is something we might understand better. I fell flat on my face when I tried to purify this receptor. It just didn't work. I struggled with it, trying to isolate it from the membranes of macrophages, and I got absolutely nowhere with it. I also used things like expression cDNA cloning, and that didn't get me anywhere either. But I was conscious of the fact that there were certain mice that were refractory to LPS. And I came to know Marina and also Chris Galanos around that time, and I talked with them a lot about the LPS receptor and what it might be, and they were certainly very helpful to me in focusing how I might find the receptor. And we kept in touch really very regularly over the next several years, and they were a part of the final study to find the receptor. I decided I would find it by positional cloning. That means cloning by phenotype. If you have a mouse that does anything recognizable, if it runs in circles, if it has no T cells, if it doesn't respond to LPS, any large effect, you could track down a single mutation responsible for that. And this was done in 3 stages. I'll come to those in a second, but let me just say we had 2 strains of mice that were refractory. One was C3H-HEJ, which is shown here. It had been around as a strain since 1965. It was also known to be highly susceptible to infection by gram-negative bacteria, and that said that you must recognize LPS to effectively fight gram-negative infection. And the other strain was less well known. It was kept by Chris and Marina. It was the C57 Black 10 SCCR strain. It had something else wrong with it too, but it was totally refractory to LPS, and I won't tell about the other thing today, too much detail. These animals had an allelic defect to the other animals. In other words, a different form of damage to the same gene. And we knew this because you could cross these 2 strains and their offspring would be refractory, even though different mutations had become fixed, one in one strain, the other in the other strain, independently. To positionally clone something, you have to first go through a phase of genetic mapping. That's where you confine the mutation to a chromosome, and I won't tell you how that's done, but it's based simply on phenotypic observation and on genotyping markers along the chromosome. We looked at more than 2,000 mice to get to a tiny area, so we felt, where we had limiting markers B and 83.3. That was as small an area as we could make the confinement, but we were dealing, we know today, with 5.8 million base pairs of DNA. The next step was physical mapping, where you had to clone all of that DNA into a contiguous stretch of overlapping pieces, and then you had to look for genes in the area because genes weren't known in the mouse. I mean, their relative locations were not known in those days. So you'd determine what the gene content of the interval was, and then you'd go sequencing all of your candidates to try to find a mutation. This took the better part of 5 years because we were in those times sequencing with X-ray film and reading the base ladders to each other, one person typing and the other reading the ladder, and you could only do a few thousand bases in a day. But we kept at it, and we were rather unlucky because when we finally did find the gene, it was far at one end, and we'd started in the middle and worked toward each end. That's sometimes how it is. I had a rather small group in those days. Alexander Poltorak and Irina Smirnova were the ones who did most of the work, but it was begun by Christoph van Uffel, who did some of the genetic mapping. And Betsy Layton helped partly in the lab in those days and also with the sequencing and organizing all the sequences. The mutation was finally found in an orphan receptor gene called TLR4, known principally for its structure. Now, the structure was very interesting, and it made a good candidate to be the causative gene involved. But I won't go through all of the details. I'll simply say that the proof of the pudding was finding the mutation, and after we saw that candidate, knowing that we were almost out of genes to look for, because almost all the DNA had been sequenced and we hadn't found any others, we identified a single nucleotide change from what normally is a C residue to an A, and that changed a proline to a histidine within the cytoplasmic domain of this receptor, and it looked like it was probably the causative mutation. But we couldn't have published that because someone would have said, well, how do you know? That may just be a passenger mutation that's crept in during the last 35 years. That was where the other allele was of key importance. In that case, we found we couldn't detect the mRNA at all, and there was a large deletion. 74,000 base pairs of DNA had been lost, including all of the exons of the gene. It was a natural knockout, and this mouse definitely had a deleterious allele. So we had 2 mice, both of them with mutations, control strains without the mutation, and this made the case that this was really the LPS receptor. Now that was exciting from the very start. We immediately began to make maps even before we had found the mutations, and we became aware that fruit flies had a cluster of genes called the toll genes, and mammals a cluster called the toll-like receptor genes, of which there were 5 or 6 known at that time, not the full complement. There were adapter proteins that might conceivably signal into the cytosol. What really amazed me was that even plants had similar genes, and plants like this flax plant, if they have a mutation in this particular member of the family, become overgrown with an infectious organism called Melanpsoralini, a kind of fungal rust. In the fruit fly, Jules Hoffmann had shown a bit before us that the toll gene, the namesake of the family, was important not only for development, as had been shown by Christian Nussland-Volhart, but also for immune defense. If you have a mutant in toll in the fruit fly, it gets overgrown by fungal hyphae. Remember, I told you that mice get overgrown by gram-negative bacteria. I thought this was too great a coincidence to ignore. Now we know the structure of lots of the toll-like receptors. They're kind of horseshoe-shaped molecules like this. All of them are dimers or heterodimers, and we know how various molecules made by microbes will bind to them, and that triggers a conformational change. And collectively, they're able to sense almost any kind of pathogen you might ever encounter in your lives, as long as it breaches host defenses. That triggers the same stereotypic response. Whether it's a virus, a bacterium, a fungus, a parasite, you get a fever, you get systemic inflammation, and this sets the stage either for much worse things or for a good adaptive immune response that can lead to long-term immunity. It was that work that led to a Nobel Prize for me, but that was 13 years in the future, and I wanted to do much more with this whole positional cloning approach, because I thought it was just wonderful that you could go from any kind of phenotype to a single base pair change and not ever make any guesses about what the target molecule was. You would just inexorably find it. The problem was that there wasn't enough phenotype in mice. You'd have to make your own phenotype in general, because there weren't that many examples of mice like C3H-HEJ or C57-BLAC10 SCCR. I moved from Dallas, where I had done this mapping work, to the Scripps Research Institute, where I could set up a bigger vivarium. And I began giving this chemical mutagen, ethyl nitrosourea, to mice. You give it to male mice, and it mutates the stem cells that make sperm, spermatogonia. And then those mice will transmit mutations to the G1 generation, their offspring. You take male G1 mice that are carriers for mutations that occur at random all over the genome. You breed them to normal black 6 females, and you get offspring where the daughter mice have half the mutations in general that came from their father. You then cross the daughter mice to the father that carries all the mutations. And at that point, in the G3 generation, you can see homozygosity for any mutation that was in the G1, so long as it's not homozygous lethal. And we used small pedigrees in those days because we were cost conscious, and we thought we can only clone things that have a very large effect size anyway, nearly qualitative effects. So let's just work with these small pedigrees. If you have just 6 mice, there's a 50% chance that you've captured any particular mutation that was present in the G1 founder. And these are all some of the phenotypes we saw. We could see quite quickly we were having an effect with the mutagen, and all of those we eventually tracked down, and some of them had immunologic effects, as well as visible phenotypic effects. We were mainly interested in innate immunity, and I kept all of the toll-like receptors under surveillance, stimulating them with their ligands and measuring TNF and interferon production as outputs. Over about 11 years using this method, we tracked down 77 phenotypes to a total of however many I say here. Is it 36 genes. 11 of those genes were new, that they hadn't been known to participate in signaling from any other means. And several of the others were very interesting, notwithstanding that they weren't completely new. They were new alleles. We found, for example, carrier proteins like 3D that were needed to escort TLRs to where they needed to be in the cell in order to signal. The same category, I would say, was for SLAC15A4, for the nucleic acid sensing TLRs. There were also adapter proteins that were new at the time that we tracked down, like TCAM1. We found mutations in a gene that was needed, its protein product was needed to process TNF itself, and so forth. All very interesting things. We saw the most exotic looking mice, like Pinky shown here, which has a form of vitamin D deficiency because of a mutation in RXR alpha. The knockout would have been lethal, but we saw this extreme phenotype, and it affected immunity as well, made a T helper 1 extremely polarized phenotype. We saw mice with spontaneous inflammation based again on a single point mutation, and here too the knockout allele is lethal. We found mice that had this abnormal distribution of fur, That turned out to be from severe iron deficiency. They had a mutation in a protease called TMPRSS6, and the function of TMPRSS6 is to cleave a certain protein called hepcidin, or to lead to the regulation of hepcidin. More exactly, hepcidin is a protein that limits the uptake of iron from enterocytes in the GI tract. And there are people with this same phenotype, as it later turned out, with something called IRIDA, for iron refractory iron deficiency anemia. That's usually the case with these mouse mutations. They're true to the human condition, and if you look hard enough, you can find the equivalent human disease. There were mice also that had movement disorders. This was an example I can show you of a mouse we called ADD, since it seemed to be constantly running in circles and not doing its homework, let's say. This we could map by positional cloning. It was a mutation in a gene coding for a new enzyme called TOM-T these days, which was a new catecholamine O-methyltransferase. The lack of this enzyme caused vestibular disease and deafness, and here too in a large series of Iranian families we found the very same kind of disease. This is a very nice one. I can't resist showing it to you. It was a mutation called possum, and I think you'll see why in a moment. One day, one of the technicians was working in the mouse house and picked up one of these mice, and you always scruff a mouse like that in order to hold it, and you do it carefully, and in this case, when she put the mouse down, it did that. And she thought she had killed it, actually, but it wasn't dead. After a second or two, it began breathing again. It has this kind of waxy flexibility, and the tail gradually falls down. So you see now it's breathing. It's actually conscious and awake. We did electroencephalography on the mouse. And I'll try to go forward a little bit quickly, because I know this lasts a long time. Well, I don't have control of that function, I'm sorry. But you would see that this mouse woke up perfectly fine if you prod it after a few more seconds, and it walks away. And you can repeat it right away. It's not refractory to a second challenge or a third. They don't seem to like it very much. They get irritated actually. But this turned out to be a mutation in a channel protein, a voltage-gated sodium channel. The mutation is right here. And normally this particular transmembrane helix, it will move under the influence of a voltage change, and that will allow sodium ions to flood into the cell. The mutation makes that particular helix get stuck at a certain position, and the cell is overflooded with sodium. This makes for a dominant phenotype, and it still isn't really understood why, but that mouse can only be triggered to do that if you scruff it, and by no other way. You can't give it any other kind of noxious stimulus. Also, the channel protein is not expressed in the brain per se, not in the central nervous system. It's expressed in the dorsal root ganglia, outside the brain. And how this all comes about, we don't know. That's for neuroscientists to work on, not immunologists like me. We saw mice with coat color mutations that created immune phenotypes as well, and I won't belabor the point, but I'll show you a spoiling slide. This particular phenotype took us about 3 years to track down, a new viable phenotype caused by mutation in this essential gene. And later we found a mouse that looks rather different, but a little bit similar, which we called muskrat. Rather than taking 3 years to know what muskrat was, we knew it immediately. At the same time, we identified the phenotype. Now, how can that come about? That's the next chapter of the story. The problem with all this wonderful induction of phenotype and mapping was it was just too slow. It was limited by genetic mapping, which meant you had to make a stock of the mice, you had to outcross to a mapping strain, wait for offspring, then backcross them to the original founder strain, then genotype and phenotype confined to a locus in the genome. And even after we had very fast sequencing technology to find which mutation was causative out of about 60 or so that exist, you had to spend a couple of years. There was nothing quicker than that. And at that, it might not be something new. It might be a new allele of a familiar gene. I moved back to UT Southwestern in 2011, and I thought of a way that we could make the process much, much faster, actually to take no time at all. And I won't go through how that works. I'll talk about it 2 days from now in detail. But I'll just say that these days, every G1 mouse gets whole exome sequenced up front, and then it's bred if it has more than 30 mutations or so, which is the minimum we like to analyze. We then look at the third generation mice, and we make very large pedigrees with about 40 or 50 mice all at once, and we genotype all of these mice for all of the mutations that were found in the G1. So in every mouse, each of 60 or so mutations is analyzed, and we tell whether it's homozygous, heterozygous, or reference allele homozygote. Having that information, a computer can use statistical computation to check every one of the mutations very fast and tell you whether it is associated with the phenotype or not by a dominant or a recessive or an in-between model of inheritance. The computer reports to us something like this: tests of the null hypothesis for every mutation in the pedigree, with one of them very clearly rejecting the null hypothesis. In this particular case, it's responsible for a certain phenotype. This would be a representation of the data. This is how homozygotes behave, heterozygotes, reference allele mice, control mice without any mutations at all. We also have a machine learning component to the program, so we have a good diagnostic. It would tell us in this case that this mutation is highly likely to be causative of the phenotype. And that program is trained by recreating the phenotypes one at a time, so that we know which mutations to look at. We don't ever look at things that are improbable. We just keep the whole colony under the pressure of mutagenesis. And if something with a low probability of verification is really true, it will be hit by other mutations, and eventually it will move up the scale, and then we'll verify it if we think it's interesting. Using this approach, we keep mutagenizing, and the computer is aware when we see phenotypes that are similar to those seen before. And it will put those mutations, different alleles, together into what we call a selective gene superpedigree. And then, even without making a CRISPR to verify or refute what we've seen before, we can have astronomical p-values, usually with, as shown by the large symbols, mutations that really create damage to the gene. So, day after day, we look at Manhattan plots that are brought to our attention and scatter plots, and we have these sharp spikes or sometimes isolated mutations on a chromosome that reject the null hypothesis of non-involvement in a phenotype. And we don't even know, I don't know what all of these phenotypes are, because each mouse is checked for about 100 different phenotypes, but we get enormous numbers of causative mutations in every screen that interests us. Here would be 400 more examples, just to make the point. As of this morning, we had finished assigning causation of phenotype to 30,072 phenotypes. We say this phenotype is caused by this mutation. That means 30,072 positional cloning projects, where the first one had taken us 5 years. To date, we've looked at more than 250,000 coding and splicing changes, which is where phenotype comes from. And we've tested more than 28 million null hypotheses in all, because each of those mutations is checked in many phenotypic assays. We've declared, as I mentioned, 30,000 instances of cause and effect where a phenotype emanates from a certain mutation. And in all, 5,726 mutations caused these 30,000 phenotypes. That tells you usually when we have a mutation, in the average case, it scores in more than one of our phenotypic screens. These mutations fell into 2,524 genes. And we know that we find about one mutation that's declared causative for every 1,000 that's examined. This new method, called automated meiotic mapping, it finds things so much faster that if we had used the old way when I was at Scripps 11 years ago, it would have taken us 1,650 years to track down the cause of so much phenotype. Where immunity is concerned, that's sort of our bread and butter area, we've found now a total of 3,784 causative mutations that fall into 1,573 genes. That's for all immunity and inflammation-related phenotypes. Our program also measures saturation, as defined by at least 2 hits in a gene where we estimate that the mutation is severely damaging to the protein, and 2 hits in the homozygous state. For flow cytometry screening, we're at 60% saturation, 60.6% actually, of all of the genes have been fully investigated, given a chance to show that they are or are not associated with phenotype. If we estimate how many genes are really required for immunity, given this level of saturation, we might say about 2,600. But we really haven't kept all of immunity under surveillance. We don't give mice tuberculosis, for example, or other infections to see whether our mutations compromise the response. So maybe there are 3,000 or more genes that are concerned with immunity. We don't really know the number, but maybe in that area, making it up a little bit. That's something like 15% of all the genes there are in the genome. Now, a lot of our work to this time had been to make new diseases, as I indicated. We found all of these new disease-causing genes, and some of them have human counterparts, others hasn't been identified yet. LMBR1L, for example, creates a new kind of severe combined immune deficiency disease, and it does so by de-repressing Wnt signaling to the point that lymphocytes undergo activation-induced cell death, and you have hardly any lymphocytes left in the case of the mouse. Hasn't been found in a human. I assume it will be at some point. The next disease is something to do with calcium signaling in B cells in particular, but T cells as well. And it isn't something that causes immune compromise per se, but if you have a mutation in that gene, in either member of a pair of genes, in fact, then you have a situation in which you have restriction of B cell malignancy growth. So maybe a therapeutic target. And I could go on down the list, but at some point we thought that this very powerful system could be turned around and used for a different purpose. We thought perhaps we can use it to suppress disease and to identify therapeutic targets immediately. There are all kinds of diseases that you might look at. You could look at environmentally induced disease, feed a mouse too much sodium, see if it develops hypertension, try to suppress that. You could use it to look at polygenic diseases like type 1 or type 2 diabetes. See if you can find mutations that suppress the development of pathology there. You could look at something like cancer as well. And we've begun to do these things. And I'll just show you a quick example. We took a kind of mouse that has autoimmune diabetes. This is called the non-obese diabetic mouse, or NOD mouse. It's known that this is polygenic, just like it is in humans. It doesn't behave in a Mendelian fashion. In the mouse, penetrance is about 80% or so. And nobody knows what most of the genes are. We know that the major histocompatibility complex is important, but not actually which gene in the MHC complex is critical. And after that, we don't know much at all. There are large regions of DNA where people have introgressed whole parts of chromosomes. And they say somewhere in this part of the chromosome is a suppressor of diabetes, somewhere is something that augments. But we have the resolution to come down to one nucleotide and say that this is the gene, this suppresses or this doesn't. On the left is an example of something that accelerates diabetes. Homozygosity for a certain mutation here makes mice develop type 1 diabetes much more quickly and to a high level of significance. This is a mutation in a protein called XPNPEP1. It's an aminopeptidase, a protein that clips the amino residues off of the protein, and it might be involved in presentation of antigens within the thymic epithelium, and it might be the case that without this enzyme you have inappropriate negative selection of T cells and accelerated type 1 diabetes, but we don't know that for a fact. Here on the right is a case of mice that have a mutation that prevents them from getting type 1 diabetes entirely. That's a mutation in coronin 1A, and the problem there is that the mice have very few T cells, and this is known to be a SCID mutation. It's mainly a proof of concept. But note that we have a total of 5 in this category, 7 in that category. If we were to go ahead with this, we'd find every influential gene that tends toward type 1 diabetes or prevents it. This all goes to the point that if we understand such things, we ought to be able to make diabetes synthetically on any kind of background. Richard Feynman famously was saying quite a lot, if any of you have read Surely You're Joking, Mr. Feynman, you know how he railed against the idea of mathematical proofs being simply memorized as to their consequences. And he told his students again and again, what I cannot create, I do not understand. And I think we have something a little bit similar in biology, that if you truly understand a biological system down to every protein that's involved, you should be able to predict how the system will behave if you damage one protein, if you damage another, if you damage both, or 3 or 4 of them, or if you augment one or the other. You should be able to predict precisely how the system will react. In other words, you should be able to make a synthetic phenotype. We're approaching that now with cancer, and this is the last I'll say. I'll just be very quick about it. We decided, let's use this screen to see if we can find single nucleotide changes that give mice robust resistance to cancer that's otherwise lethal to the mouse. We kept the mice under surveillance, as we always do. After a few hundred pedigrees went by, we found a strong resistance mutant that we called icebreaker. And after a few hundred more pedigrees went by, we found a second mutation that also is significant, and it too causes elite resistance. This we called citation. Now we know that icebreaker allowed the formation of a new kind of CD8 T cell receptor that normally gets eliminated by negative selection. And that was how it worked. That T cell receptor, or group of T cell receptors with that specificity, will destroy almost any tumor that you give to C57 black 6 mice. Citation affected the priming of CD8 T cells by a completely different mechanism, and it made sense to try to combine these 2 mutations. Both are such elite controllers that you can't really tell an advantage if you have 2 of them in a mouse when you give it a subcutaneous tumor. But if you give it intravenous cancer, if you give melanoma cells IV to the mouse, then that's also assuredly lethal. And it's such a stringent model that you can see the contributions of the 2 mutations. In this picture, I'm showing you how mice behave without checkpoint inhibitor or with checkpoint inhibitor. A wild-type mouse, shown by the black symbols, will almost always die by 30 days after you inject melanoma into it, whether you give a checkpoint inhibitor or not. That's similar to humans. Usually checkpoint inhibitors don't do anything. Only in the lucky 30% of patients or so are they beneficial with melanoma. The icebreaker mutation isn't protective by itself. Maybe citation is slightly protective, but it's marginal. Combine the 2 and you have a significant protective effect. rather than either of the mutations independently. And this difference is greatly amplified if you give anti-PD-1, whereas with no checkpoint inhibitor you have no, with checkpoint inhibitor in wild type you have no effect. Each of the mutations is better able to work in the presence of checkpoint inhibitor. Now we have 10 mutations at this point. If we add a third, a fourth, a fifth, will we eventually be able to make the mice absolutely resistant to cancer? That's all in the works, and I can't say for sure. All right, last slide, I think. What have I learned from all of this story that I've been telling you? How I started out very interested in nature and wound up with a Nobel Prize doing things that are just fun for me to do. I'll say that it's good to have a strong motivation and to start young. I was very lucky in my case that I had a father who was in biomedical science, and he was very supportive of my learning more about it. But that was what drove me, and it still drives me today. I think it's just amazing that molecules make such a complex phenomenon as being alive. I studied medicine, and that undoubtedly helped me also. Maybe for some of you, you've gone the direction toward the PhD route, and I would say that not all is lost, but it did help me anyway to study medicine. In science, most of us are taught that you have to have a hypothesis, and this is the key thing. And I would disagree with that. I think that hypotheses and teleologic reasoning, those are overrated, and they can often mislead you, especially if you're prone to bias, and almost all of us are. It's really important to be skeptical if you're a scientist, and foremost, be skeptical of your own data. Really be resistant to being convinced that any hypothesis you make is correct. And you can get away making no guesses, as that's what I usually do these days, using ENU and just tracking things to a mutation without guessing. But if you do use hypothesis, then by all means take R.A. Fisher's recommendation seriously and just give the facts a chance of disproving the null hypothesis that you've made. Working hard is important, and I've always worked very hard, and I regret that these days it looks like people don't work as hard as I used to work when I was young. But I say to people, you should work hard. You're in a competitive business. Work hard and don't apologize for it, but work on something you love. Work on something that you find compelling and exciting so that you're motivated to come to the lab early every day and work all day and know what you want to do and get things done and keep a list and check things off. And then you won't have to be asking questions about work-life balance because your enjoyment in life is your work. Finally, I have to say you've got to be very honest if you're a scientist. And this applies with regard to your work, not whether you cheat when you're playing a game of poker, let's say. That's all right with me. But if you are working in the lab, you should not exaggerate your results. You shouldn't mislead people in any way, in your own lab or in the world at large. If you find you're doing that, then you're in the wrong business. You're not in science for the right reasons. And this is something really unique about science. I would say in some professions it's good to be dishonest. If you're a magician, for example, you have to mislead people, and that's perfectly acceptable, and a good magician can't be a good magician without doing that. I suppose the same is true if you're an actor, or perhaps if you're a politician. And it's not true in science. We're playing very different games. And with that, I want just to make one more comment. That I didn't do all this alone. I have a large lab now. These people work as part of a team. Some of them create mutations, some breed the mice, some genotype the mice, some do the phenotypic screening. There's an excellent computational group that made the software that does this magic that I showed you and speeds us up so much. And there are those who do CRISPR-Cas9 confirmation to train the machine learning program and to make us sure of our results. And finally, I've started a company because I think we've pushed this as far as we can in a laboratory setting. We could be mutagenizing on a much larger scale, finding resistance to all kinds of phenotypes, disease phenotypes in the mouse, and having therapies. Phenogenetics was started to accomplish that. And I'll be delighted to talk to any of you, answer your questions, and tell you anything more I can now. Thanks.